Welcome to another episode of the Synthetic Biology series. We are NTU-Singapore, and today we will explore the world of drug discovery using synthetic biology. Drug discovery is one of the most crucial processes in the fields of biotechnology and pharmacology. It is how new candidate medications are discovered, analyzed, and utilized to treat different conditions. Before we can appreciate the use of synthetic biology in drug discovery, let us try to identify and understand what synthetic cells are. These synthetic cells are used as biofactories to manufacture chemicals that are often challenging to synthesize by traditional pharmaceutical industry processes. A typical synthetic cell is composed of three components. An inducer gene is connected to a genetic circuit that produces an output signal based on the inducer gene, and a reporter gene at the end to display the output signal. These synthetic cells can be integrated into hosts to facilitate gene expression of targeted compounds. These biosensors can be used in many ways, for example, to validate drug targets, understanding a drug's mechanism of action through a designed disease model, and induce a drug delivery mechanism at a specific site or under specific conditions. We will now see some examples of how synthetic biology principles are used in drug discovery. The development of genetic circuits helps in increasing the signal flux of secondary metabolic pathways. The concepts of protein engineering can be employed to explore the chemical diversity of secondary metabolites. Similarly, optogenetic biosensing can validate drug targets and monitor drug delivery in the host cells. This illustration shows the many applications of SynBio in drug delivery. The critical point to note is that SynBio has made itself an indispensable tool for modern drug discovery and has paved a path for significant progress in the field. Now, let's try to understand the importance of SynBio in drug discovery by looking at some isolated examples. We are all aware of the effects of the COVID-19 pandemic, but most of us aren't mindful of the contribution of SynBio in finding solutions. Without SynBio, we wouldn't have been able to develop COVID-19 vaccines so quickly, nor would we have rapid testing kits. Let's try to understand the technology behind this. Today, we have the world's first automated DNA printer, the BioXP3250 system, designed by Codex DNA, which allows rapid and accurate printing of long stretches of DNA that can be automatically assembled into a synthetic gene. This is what it looks like. The system was used in making the world's first SARS-CoV-2 synthetic genome. The genome is now being used by pharmaceutical, vaccine, and diagnostics industries. The template is used to identify the optimal active ingredient for COVID-19 vaccines to generate the best immune response once administered. Researchers have been using the template to create antibody-based drugs that might provide resistance against COVID. The diagnostics industry uses the genome to identify regions that can be used for rapid testing and develop testing kits that are sensitive, accurate, and quick. Now, let's look at another application of SynBio in drug discovery. We shall look into immunotherapy, a new method for the treatment of disease by activating or suppressing the immune system synthetically. Before we talk more about immunotherapy, we must first understand what T cells are. T cells are a part of the immune system that focuses on specific foreign particles. T cells circulate until they encounter a specific antigen. As such, T cells play a critical part in the immunity to foreign substances. Immunotherapy is currently being used to find a cure for cancer. T cells are being modified using SynBio to become more sensitive to recognize and fight cancer cells. The process of immunotherapy involves engineering cancer-fighting T cells. First, the T cells are removed from the host's blood and taken to the lab. gene encoding is then carried out. These T cells are modified to engineer T cells with specific receptors that can bind to the required antigen. Therefore, the surface of these cells are covered in chimeric antigen receptors, CARs in short. Modified T cells are further harvested and grown in the lab. Finally, the bioengineered cells are infused back into the patient. Now the patient has antigen receptors that can identify and eliminate infected cancer cells from specific regions. Although the process looks pretty straightforward, it is met with a lot of hurdles. Different tools, gene circuits, and synthetic receptors can tackle some of the challenges faced by immunotherapy. The challenges are primarily associated with safety, efficacy, and manufacturing difficulties. Receptor engineering helps in designing synthetic cells which are reprogrammable with user-defined input-output signals. This can help to achieve modified T cells which can be controlled better. The binding of CARs to the T cells is not always perfect. Sometimes CARs or T cell-related toxicities can come up, which can lead to tumor lysis syndrome. This can be controlled because of the advancement in the SynBio field, which allows us to manage and mitigate toxicity by designing safer CARs. Another example of the application of SynBio in immunotherapy is the advancement in control over the efficacy of CARs T-cell therapy by designing newer gene circuits and/or CARs. From the examples, it is clear that there is a strong relationship between SynBio and drug discovery in terms of finding a solution to diseases that involve gene mutation. Therefore, the advancement of SynBio is crucial for drug discovery against unknown pathogens and antigens. Thank you so much for listening, and we hope that you have learned something new about synthetic biology.